It seems like we're in good shape to go. Welcome back, everyone. It is our second session of the Leading Edge Symposium. We have an amazing lineup of biochemists for you. And my only job here is to just introduce our two amazing chairs, Clarissa Dury and Lam Q Fong, who will take it away from here.
Hi, I'm Clarissa Dury. I'm a postdoc in Melanie Oi's lab at the University of Michigan Life Sciences Institute, and I'm a 2020 Leading Edge Fellow.
Good morning. My name is Lam Q Fong. I am a postdoc at UCSF in the lab of Bill DeGrado, and I'm also a 2020 Leading Edge Fellow.
And just a quick reminder, the talks today will be 15 minutes, and then we'll have 5 minutes after each talk for questions and answers. So if you have any questions for the speaker, you can raise your hand using the reactions in your Zoom window, or you can type your question into the chat box. And you'll also note that these talks are being recorded.
Okay, so let's just get started. It is my pleasure to introduce our first speaker, Lisa Eshun-Wilson, who is a 2021 NSF postdoctoral fellow in the lab of Gabe Landers at the Scripps Research Institute. She also just was awarded the Ford Foundation Postdoctoral Fellowship, and today she'll tell us about her work on MAP7. So I'll let Lisa.
Awesome. All right, let's get started. Let's see. Okay, wonderful. Thank you. Can you guys hear me okay? And see my slides okay? Okay, awesome. Thank you so much for that introduction. It's such an honor and pleasure to be part of this fantastic community. I really want to take a second and just thank Kara for organizing this fantastic group and this fantastic symposium. It's an absolute honor to be a part of this today. So the title of my talk is the effects of microtubule-associated protein 7, MAP7, and molecular motors, or specifically on molecular motors and intracellular trafficking. And so throughout my postdoctoral or my pre-doctoral work, actually, which I'll be focusing on today, I was very interested in the tubulin code. So how cells are able to read and write unique microtubule populations. And so I was particularly excited about MAPs, or microtubule-associated proteins. And today I'll focus on MAP7. So since I was an undergraduate, as a biochemistry major, I got extremely excited about the core principle that form follows function, and that structure is a great tool. Structural biology is an excellent tool to provide insight into an enzyme's role in the cell. And so my first project in graduate school focused on how a small chemical modification could have a dramatic change on the global stability of the microtubule. And second, I built upon that experience to now look at how individual microtubule-associated proteins were not only affecting microtubule stability, but each other and motors in microtubules as well. And finally, I've built upon this work to now look at multimodal enzymes and how they make molecular decisions now in my postdoctoral work. But for today, I'll be focusing on the story of MAP7. And so the core team behind this work was Eva Nogales and Chengling Fang in the Nogales lab, who's now faculty in China. And then Ahmed Yildiz, another fantastic faculty member at Berkeley, and especially Luke Farrow, who was my absolute core teammate throughout this work. And so microtubules are made of alpha and beta heterodimers that stack head to tail that associate laterally, which I'm showing here, to form a hollow tube in the presence of GTP. And their stability and overall function in the cell is essential for a myriad of different properties and functions within the cell. And so microtubule-associated proteins are really critical for stabilizing microtubules and regulating their roles in the cell. And here I'm highlighting, for example, MAPs that can stabilize microtubule structure. We also see that there are MAPs that can form these ultrastructures in the cell. They can create microtubule bundles, for example, or space them at a very particular distance to form these very beautiful arrays of microtubules. And then finally, we also see that MAPs directly regulate motors. So kinesin and dynein are highlighted here, two distinct motors that are essential for intracellular trafficking within the cell. And so our lab, the Nogales lab, was very interested in tau in particular. And so the misregulation of tau can lead to life-threatening diseases, one largely known as Alzheimer's. And so here in this image, I'm highlighting a healthy neuronal cell that's being decorated by MAP7, shown in purple, and it allows for it to form its critical structure. Whereas in an unhealthy cell, in particular a cell that now has that is suffering from the misregulation of tau and leading to these neurofibrillary tangles, as they're called, of tau, we're seeing now that the cell is unable to form critical microtubule structure, ultimately leading to Alzheimer's. And unfortunately, you can see the microtubule starting to completely fall apart. And so the molecular mechanisms of how tau was binding the microtubule to stabilize it was really important and interesting to us. And so when I was a third-year graduate student, we were able, our lab was able to resolve the high-resolution structure of tau, which provided exciting answers, but also sparked new questions. So here I'm highlighting tau in purple that is decorating the microtubule along the protofilament crest, so the central region of a protofilament, and in order to stabilize longitudinal contacts. And then what I'm highlighting here is the first author, Elizabeth Kellogg, was able to now build the entire structure of tau, particularly the microtubule binding domain, and provide insight into where it's phosphorylated, which is critical for its regulation in the cell. And so as this work came out, I had the chance to attend a really fantastic meeting in Heidelberg, Germany, where I saw a phenomenal talk by Cassie Ori-McKenney, who's a professor at UC Davis. And she talked about how microtubules, and microtubule-associated proteins, along with motors, all had this really interesting connection in terms of their regulation and competition between each other. So here I'm highlighting a microtubule now in gray, and you can see two different motor proteins highlighted in green. And then the microtubule-associated proteins are now in this dark orange. And specifically, this is their microtubule binding domain. So what I want to draw your attention to are these really dramatic projection domains. And what really stood out to me was MAP7. It had the largest projection domain. And what Cassie showed was that actually MAP7 could compete tau off of the microtubule lattice, and it could recruit kinesin to the microtubule lattice. So it had this really interesting biphasic property within the cell that I got really excited about, because MAP7 seemed like another tool to understand more about tau's function in the cell. And then also, because we had an understanding of the microtubule binding domain, it also seemed like a really nice way to learn more about the projection domain within the microtubule, or within the projection domain and its effect on the microtubule. And so I ran back to Berkeley after this phenomenal, I would say, meeting that really opened my eyes to the potential role of MAP7. And so I got excited about what are the molecular mechanisms between MAPs and between MAPs and motors, and how are these potentially exploited in disease states. And so what we knew was that MAP7 and tau were competing to bind microtubules, and that MAP7 had a higher affinity for microtubules. And that might have been a way that it is potentially competing tau off of the lattice. But how were MAPs competing? What were the molecular mechanisms behind this competition? We also knew, especially from Cassie's work, was that MAP7 recruits kinesin. It's the only MAP to do so via its kinesin binding domain, highlighted in pink here. And then both MAP7 and tau inhibit dynein. So there's this really interesting play here. MAP7 can inhibit dynein but recruit kinesin, and I was really wondering why. So the questions this sparked were is the interaction between the MAP and kinesin direct? And then how do the MAPs regulate motor-driven traffic, for example? And so the large model in the field was that MAPs compete for the same binding site. So the proposed idea was that since tau is binding along the microtubule, specifically at the protofilament crest, MAP7 must also bind there, and that's how it's able to compete it off. However, there was no structural information about MAP7. So I set out to resolve the first structure of MAP7. And thankfully, with our fantastic collaborative experience, we were able to see and visualize the first structure of MAP7. So immediately what stood out to us was that MAP7 is binding at a novel site and potentially stabilizing microtubules in a novel way. We also noticed that there were two binding sites. So one I'm showing here where you see direct density showing that MAP7 is contacting the microtubule lattice, whereas here where you see the absence of a density and indicating that it's a potentially weaker binding site, which I proposed could potentially be a site that can accommodate kinesin. As MAP7 is present, kinesin also still has space to walk along the microtubule and pool its cargo of interest. Which was really interesting to us. And so this sparked a new model, a novel mechanism for MAP competition, because what we propose is that MAP7's microtubule binding domain or its projection domain is driving MAP-MAP competition. And so together with Luke, I was able to test different constructs in order to answer this question. And so first what I'm showing you is a single molecule biophysics experiment, a fluorescence experiment, where in this channel, you're looking at tau, decoration of the microtubule. And here is, you have the microtubule labeled in this channel. And so without any MAP7 present, we see that tau is able to fully decorate microtubules, which we expected. And then once we added MAP7, we now start to see that MAP7 is now decorating the microtubule lattice and kicking off tau along the overall microtubule structure. But interestingly, when you now add just the projection domain of MAP7, you now start to see it also tau getting kicked off. And so just the projection domain, no microtubule binding domain, you're still able to kick off tau. And so to show that it was the projection domain driving this competition, we also wanted to test if the microtubule binding domain alone could co-bind in the presence of tau. And so that is exactly what we saw. So when we added tau, and you're looking at tau decorating multiple microtubules now, and then just the microtubule binding domain of MAP7, you could now see them co-binding, indicating that the projection domain has a driving effect on the competition. But on a molecular level, we really wanted to see, ideally, a co-structure of MAP7 and tau to really drive this point home. So I set out to solve that structure and thankfully was able to do so. So we could confidently say that MAP7 binds at this novel site on the microtubule, and propose that MAP7 and tau competition is now driven potentially by the projection domain of MAP7. And so could this be a way for the cell to fine tune MAP7 as a means to regulate tau? And so now I'll show you a summary movie, just highlighting what I've shown you so far. Here we have full length MAP7 decorating the microtubule lattice. And now we have the microtubule binding domain of MAP7 decorating the microtubule in the presence of tau. And so we could start to paint a picture of how MAPs are regulating motor guided transport as well. So although MAP7 recruits kinesin and tau inhibits kinesin, both MAP7 and tau sterically block the motor binding site. So here I'm showing MAP7 and kinesin overlapping at the same site, as well as diamine, and also kinesin overlapping and now diamine. And so what we propose, or our model, is that kinesin must interact with the projection domain of MAP7 to walk along microtubules. And so I'll show you a schematic of what I mean. As we potentially increase the concentration of MAP7, what we're proposing is that under low conditions, MAP7 can directly interact with its MAP7 binding domain within kinesin via its kinesin binding domain. Here it's highlighted in yellow. And then as you increase the concentration, you have this tethering effect by the projection domain, keeping kinesin present on the microtubule lattice. But at higher concentrations, you start to see this steric overlap winning in terms of this favorable interaction, winning over that favorable interaction. And so you start to get inhibition by the microtubule binding domain. So this sparked this new potential model for motor regulation by MAP7. And so our take-home messages and future directions is that MAP7 binds the microtubule in a novel location, which provides insight into how MAPs influence each other and other motors. And cells may use MAP7 to regulate tau and microtubule stability, underscoring its relevance in disease. And then questions that still remain are how does the protection domain recruit kinesin, and what are the molecular mechanisms of this process? And then what types of interactions occur between kinesin and the kinesin binding domain of MAP7? Because the projection domain is so flexible, it remains a challenge to be visualized. And so this work, I would say, really excited me about studying multimodal on very versatile enzymes and how they make overall molecular decisions. And I believe this, my pre-doctoral work as well as my postdoctoral work has set me up really nicely in my future work to now combine training that I'm getting in cryo-EM and soon cryo-ET to explore the role of particularly deubiquitin enzymes, or DUBs, which are serine proteases, and their role in nutrient transporter turnover at the plasma membrane. So personally, this system is very interesting to me because you have this turnover of very critical nutrient transporters. And what you see are that you have deubiquitin enzymes that are playing critical roles in deciding which cargo to protect. And I'm really interested in how that decision-making takes place and how it changes under stress conditions. And particularly what changes regarding its substrate specificity. And so with that, in my last minute or so, I would just love to acknowledge all of my fantastic mentors throughout this process, and especially Eva Nogales, who has really been a beacon throughout my training and my graduate career. And now my postdoctoral mentor, Gabe Lander, who is setting me up well for my future interests as well. So I'll stop there and happily take any questions that you guys have.
That was a fantastic talk. Thank you, Lisa. So make sure you raise your hand for questions. Give it a second here.
Thank you so much.
I can start off with a question. I was just curious, because so here you're looking at the isolated effect of MAP7 and how other MAPs might play into roles, and whether the binding site that you found was associated with, like the alternative binding site you found was associated with a different MAP.
Yeah, this is such a great question. So this is the first time we see a MAP bind in this site, which was very exciting for us. But you're absolutely right. There are many more MAPs that need to be resolved. And in particular, MAP7 and tau are part of a family that we call structural MAPs, because they play such a huge role in regulating ultrastructure of microtubules throughout the cell. And so to answer your question, when I presented this work recently also at Utah, I had a very similar question. And one idea that was proposed was looking at starting off, like taking a reductionist approach, because you're absolutely right. The human context of MAPs would be really, I would say, very, very complicated. And so I would say a first step would be looking at a system such as C. elegans, for example, that has that has fewer MAPs. And so, and ideally looking at all of them present on the microtubule at the same time. That would be an absolute dream. But I think cryo-EM is not going to be enough to answer such a question. What's going to be key is looking at or using tools that have larger scales, so you can see these larger complexes in place at once. So cryo-ET, for example. And there's a fantastic professor at Utah, Eric Jorgensen, who is doing some fantastic work. Just really fantastic work, even sort of playing with the scales in terms of being able to visualize all of the interesting players at the same time. In different systems. So he's looking at synaptic vesicles, but I think that tool would be really nice to start to look at all these different MAPs at play. Because one thing I thought was that MAP7's unique binding site, maybe that would change in the presence of tau. and it didn't in the presence of tau, but maybe in the presence of another map that has a higher affinity for that spot, or has another interesting role, would change its spot, for example. Because also what we see are that these maps are very sticky and highly electrostatic. And so I wonder if there's a bit of some randomness in terms of where it's sticking, because you have this, the microtubule surface in terms of charge can be seen as highly negative in some areas. And then you have these highly positive, sticky maps that I wonder if there's many, they're just kind of sticking everywhere, which would be really interesting to see, and also very beautiful in terms of visualizing the randomness, but also the precise coordination that goes, because I would imagine that the map decoration changes for the type of microtubule population that you want to craft. Yeah, so great question.
Awesome, thank you. So here we have a question from C. Bartman. Do you want to unmute yourself and ask the question, or?
Sure. Hey, awesome talk.
Oh, thank you.
So I don't know very much about microtubules at all. So, but I was just curious. I've only heard about tau in the context of Alzheimer's. I wasn't familiar with the role it played in healthy cells, but I was just curious if it's thought that the pathology in Alzheimer's is related to effects on like destabilizing microtubules, or is it due to the like tangles themselves more?
Yeah, this is a great point. So I think in terms of timeline, what happens is for the tangles in particular, you have misregulation of the phosphorylation of tau. It starts to form these tangles, and they're sticky and they stick with each other. And especially now that they're not being phosphorylated correctly, they start to stick with each other. And then microtubules don't get the level of stabilization that they need and start to fall apart. So I would say in terms of timeline, first it's forming those neurofibrillary tangles. And then what a downstream effect is that you have deterioration of this critical structure of microtubules. And it's really interesting because you would think that since there are so many maps that are playing in stabilizing roles as well, for the microtubule, you would think, okay, if tau's gone, and it's completely messed up, why can't another map kind of step in and help stabilize it? It's something I've thought about. And with tau, it's just extremely abundant. I believe 80% of maps in neurons, a large percentage of maps are tau. And so we just don't have that same level of abundance in terms of being able to stabilize microtubules in the same way tau does. So if tau is gone, because it's such a core player, you just don't, the backup players that are coming off the bench, they just can't fulfill that role. Yeah, I mean, yet, and that's based on our understanding. So I think that if we understand more about how maps are switching roles, and which goes back to the first excellent question, then we can think of potentially using another map, maybe structurally engineering it so that it can replace tau in such a way that is significant enough. I'm of course getting ahead of myself. I'm like, we can solve Alzheimer's in our lifetime. Of course we can. But I think this is a really important question, and very, very interesting because yes, so if we could engineer another map to perform as significantly as tau, that could be a way to offset tau's absence. Yeah.
Thanks.
Thank you so much, Lisa. I think we're going to go on to the next speaker, but there's a few more questions in the chat if you want to take care of those.
Oh, yes. Thank you. And thank you guys so much. Again, such an honor to be a part of this. And I'm so excited to continue to get to know everyone. Thank you so much.
Thank you.
Thank you. All right. Our next